online sessions of Pharmaceutical Microbiology. Myself, Ms. Tosha Pandya, and my colleague Ms. Kaushika Patel of Department of Pharmaceutical Technology will be demonstrating the practical tutorial of microbial assay of antibiotic. In this practical, we will see the microbial assay of antibiotic by cup plate method. So what is microbial assay? Microbial assay is a technique with which you can find the potency of any antibiotic or assay of any antibiotic and directly on the find the effectiveness of antibiotic on a particular microorganism. For each microorganism, you will find the IP monograph in which you will find the all conditions which need to be followed for a particular practical. So now, what is the principle behind the cup plate method? Cup plate method, as it suggests, it is performed in the Petri plates with the help of solid nutrient agar media. It is based on the diffusion of antibiotic through a solidified nutrient agar media which is previously treated with a particular microorganism. So what all you require is a standard solution set of the standard antibiotic solution whose potency is already known from S1 to S5. Here we will take 2, 4, 6, 8, 10 microgram per ml of the standard solution in which you can see that the S3 is the median concentration. Now you have to prepare 5 different Petri plates. Along with the standard solution, we will also have an unknown solution whose potency need to be found. Okay? So you will take 5 different Petri plates in which you have the nutrient agar which is previously seeded with the microorganism. Here for gentamicin you will take S. aureus. So they are seeded with the S. aureus. You will form 4 different bores in a particular plate with the help of the borer aseptically. This technique should be performed aseptically. Now in the bores which are formed, you will add the respective antibiotic solution in this pattern in which you have alternate cavities of particular dilution with the median concentration from S1 to S5 and unknown. And now the equal volume should be added of the antibiotic solution. After this, you will incubate for 1 hour in the refrigerator so that it will allow the diffusion of the antibiotic solution. Finally, incubate in the incubator for 24 hours. After the incubation, you will observe that around the cavities in which you have added the antibiotic solution, you will see a zone of inhibition which is a transparent layer around the cavity which indicates the inhibition of growth of antibiotic due to the diffusion of the antibiotic. It will kill the microorganism in the surrounding layer. Now as you go on increasing the concentration of the antibiotic, obviously the zone of inhibition will increase. So you will plot a graph of log concentration versus zone of inhibition which will be a straight line graph. Now for the unknown you know the zone of inhibition and not the concentration. So from the zone of inhibition you can extrapolate and find the log concentration, take the antilog and find the concentration. The practical part will be demonstrated by Ms. Kaushika Patel. Thank you.
Hello dear students, so now we will start with the practical demonstration of microbial assay by cup plate method. So this is a working area, aseptic area between the burner in which you have to work. So let me introduce you to all the requirements which you will need. So first of all, this is the stock solution, antibiotic solution of 1000 microgram per ml, from which you have to prepare the subsequent standard solutions, that is S1, S2, S3, S4, S5. This is the U, that is unknown sample of whose potency you want to determine. This is a culture which you have to inoculate on the solidified culture medium in the Petri plate. This is a waste beaker in which all the waste material is to be thrown. You will require a micropipette to transfer the antibiotic solution into the Petri plate. This is a marker with which you have to label all the sections on the Petri plate. This is the cup borer which you have to use to dig the bores in the Petri plate. Okay? Now, between the burner on a working tile you have to work. For that you will require the cotton pads soaked in IPA. So these are the cotton pads with which you have to clean the plates. Now the Petri plate which you have to use, you have to label like this as you have been shown in the theory. So 4 sections you have to make. S3 is your median concentration, S1 is your first standard concentration. So S3 is to be used in all the plates. So in the first plate there will be S1, S3. In second plate there will be S2, S3. In fourth plate there will be S4, S3. In fifth plate, there will be S5, S3 and then there will be unknown and S3. Like this, you have to label all the plates. Okay, so now this is the cup borer which has been already cleaned with alcohol. Before you perform the practical, you have to again sterilize it by the physical method, that is by heating. Okay, allow it to cool for few seconds. Then dig the cup borer in the center, okay, and throw the waste material in the waste beaker. Same way, dig the second cup and throw the waste in the beaker. Like this, you have to make 4 cups in the Petri plate. Similarly, dig the boards in the remaining 4 Petri plates. Okay? Now, according to the label, you have to pour the antibiotic solution into the plate. For that, we will use the micropipette. Okay? You have to adjust the quantity of the micropipette. Make sure that the bore should be filled with antibiotic solution, but it should not overflow from the bore. So, first we have to take S1. Take the solution like this from the test tube and pour it in both the cups of S1. Now, take median concentration S3 which you have to use in all the plates. Okay? So, this is the median concentration S3 and you have to add it in the remaining 2 boards of the plate. Again sterilize the test tube, take the solution, flame it and keep it on the rack. Add S3 solution in both the boards and the first plate is completed. Like this, fill the solutions in the boards of all the remaining 4 Petri plates as you have been explained in the theory. In the last plate, along with the S3, you have to add unknown solution. So, any unknown solution of any concentration you can add, and based on the zone of inhibition of that unknown solution, you can determine its concentration. Okay? After this is done, you have to insert a filter paper in between the lid and the plate, bottom plate. Place that plate in the refrigerator for 15 minutes to allow the diffusion to occur. After 15 minutes, remove the plate from the refrigerator and place it in the incubator for 24 hours at 37 degrees Celsius. After 24 hours, observe the plate for the zone of inhibition, measure the 3 diameters, take the average and find out the corresponding zone of inhibition for S1, S2, S3, S4, S5 and unknown solution. Okay? Thank you.
Please do like, share and subscribe our YouTube channel Pharma Ignite and do not forget to press the bell icon to get regular notifications so that you do not miss out any of the topics in this chapter. Thank you.